next speaker is Giuseppe Gallitto with Towards Generative AI-based fMRI Paradigms, Reinforcement Learning via Real-Time Brain Feedback.
Okay, thank you for the introduction. And I will talk about our new approach called reinforcement learning via brain feedback. So, I have nothing to disclose. Let's talk briefly about traditional task-based fMRI studies. In these studies, what we do is create an experimental paradigm that has some psychological behavioral validity in order to infer the underlying neural correlates. This is the traditional approach, is what we all use, but it faces a reverse inference problem. So we have no information about the specificity of findings that the task at hand. If we show a stimulus and some brain areas activate for that stimulus, it doesn't mean that those areas are specific for the elaboration of that stimulus. What we propose here is an alternative. So instead of going from the experimental paradigm to the neural correlates, we do the reverse. We go from the neural correlates to the experimental paradigm. And what does this mean? This means that we generate or find the optimal stimulation or paradigm that maximizes or minimizes the activity in one region, a response from a set of regions, or maybe also the connectivity within a network. And we do this with real-time fMRI and reinforcement learning in order to generate a paradigm space from scratch, for example, via generative AI, or by modifying the characteristics of a pre-existing paradigm space. And actually the latter is what we do here, because here we present a very, very simple proof of concept, so no fancy generative AI models. And what we do here is we use real-time bold response in the primary visual cortex, and we feed it to a reinforcement learning algorithm that is a very, very simple soft Q learner, so a very simple textbook reinforcement learner. And what this does is just changing the contrast and the frequency of a flickering checkerboard. So here our paradigm space is just given by all the possible combination of contrast and frequency. This loop you see in the images continues till the reinforcement learner is able to find the right combination of contrast and frequency that maximizes the activity in the primary visual cortex. So basically, it has to figure out if it has to go toward the maximal contrast and optimize the frequency for each participant. To test our approach, we performed hyperparameter optimization, both using computer simulation and empirical data. So regarding the simulation, we define a ground truth that is just a linear function of contrast and frequency that peaks at maximum contrast at 7 Hz frequency. And basically the reinforcement learner will walk the paradigm space, and when it goes around the maximum contrast at 7 Hz frequency that you see in the image, will get higher reward. And this goes on for all the sets of hyperparameters, and for 100 trials till we get the optimal parameter. We also optimize in the scanner we're using with the first participants, and what they are presented with is just a simple block design. They have a fixation cross at the center of the screen, and then 5-second stimulation during which they see the checkerboard with a specific combination of contrast and frequency, and 11 seconds of resting state in which they see only the fixation cross. We take the data from each block and give it to the reinforcement learner as a reward, and the loop continues as I said. The parameters that you see on the bottom are the parameters we found to be important for our approach to work, and it's basically the SNR, so it has to be over a certain level, the smoothing of the Q table, the learning rate, and the temperature. To bring our approach to the scanner, we implemented a real-time software and dashboard that you see here, and basically on the top left you see that we acquire a functional reference volume for preprocessing and motion correction purposes. Right next there is an overview of the current volume that is rank harmonized to avoid global intensity effects, and you also see there is a mask, that is the mask of the primary visual cortex we use to extract the data. right next there is the Q table that is initialized as constant, obviously, and it contains the importance of all the combination of contrast and frequency for the reinforcement learner at the current time point. While on the bottom, you see our data that is right now flat because there is no incoming data to the dashboard. But you see our hypothesis function, that is what we fit to the data in order to calculate the reward for our reinforcement learner. So how does the process work? A random combination of contrast and frequency is selected and shown to the participant. That is what corresponds to the red dot you see on the Q table. We get the data for that, we fit it with the hypothesis function, and we get the reward for the reinforcement learner that chooses a new combination that corresponds in this case to the green dot. And this process goes on for each block. So you see that basically walks the paradigm space, changing the importance of each combination of contrast frequency, till it basically understands that it has to maximize the contrast, no, and the frequency to a certain level in order to get the highest activation possible. And this brings us to our result. We rerun the simulation with the optimal parameters and also scanned with the optimal parameters 10 healthy volunteers for 2 runs each and 35 to 45 blocks each with a TR of 1 second. So each scan is very, very short. It's around 10 minutes. And you see the results first on the matrices. In the matrices you see the Q table with the average steps walked by the reinforcement learner in order to find the right combination of contrast and frequency. On the side, you see 2 plots. There are the reward and the convergence, where the convergence is calculated just like the Euclidean distance between choices in the paradigm space. And you see that we are able to increase the reward bit by bit, but not that much, and it's normal because we have a very low running rate and there is still some exploration even at the end of the 10 minutes, but the convergence goes down very, very quickly. So it converges toward the maximum contrast very, very quickly. And the same happens for our participants. What you see on the bottom is a mean of all the runs of all our participants. And the colors of the matrix, I forgot to say, is basically the last matrix, so where the algorithm stops. So you see that it optimizes for high contrast and high frequency. Yeah, in 15 to 25 trials, we're already able to reach convergence. And here there are a few matrices at the last trial for a few runs, so you see that the result more or less is the same. But the point here is that this technique is robust on the single subject level. So we don't train a model and then use it to go over the data of each subject, but we train the model from scratch for each subject and each run. So that it finds basically the combination, or that is tailored for the participant inside the scanner. This is at circa 10 minutes, so the last trial, but you can also see that at the 15th trial, already it converged toward high contrast and is still maximizing the frequency. So it's very, very fast at converging. And the main point here is that with this method, this is a very simple example, but with this method you can already see some results on the single subject, while with a traditional method you will need a group level analysis. Okay, where do we want to go with this? We want to extend first the proof of concept. We're using retinotopic mapping, so we want to boost a bit in complexity in order to calculate better rewards and in general optimize the training protocol. So instead of having contrast and frequency, we'll have polarity and eccentricity, and we will see if we'll be able to optimize for different parts of the visual cortex. Plus, we want to test the feasibility of using generative AI, so by generating stimuli, the paradigm from scratch. And here we have some options. We could, for example, identify stimuli that elicit relevant responses in patients. We could, and this is one thing we're very fond to do, to try at least, is generate music for pain hypologisia, or we can just do reverse inference to test the specificity of various region function mappings. And we can plan to do that, or at least to try open source models, but we can also consider to use some more famous and known models. Thank you everyone for being here. I thank you my supervisor, Tomasz Pisak, and all the members of my lab. Thank you everyone.
Thank you. Is there any questions from the audience? Maybe did you want to ask in person? If not, I have a question for you. So do you think we could go farther and maybe generate stimuli that will look into functions that haven't been explored yet?
Yes, I mean, the challenge here is to define the paradigm space, because even with generative AI, it's not like you take a model and say, okay, just run, but you have to define those latent components that will generate the possibilities, of to get the stimulus. So yes, we can, but the study that has to be behind it is how to define the paradigm space. Yeah.
Okay. Thank you so much. Round of applause.